We are live now. Good evening, esteemed faculty members and all the delegates. Thank you for joining. This is Sahiti on behalf of Shield Connect, welcoming you all for today's webinar, Comparison of Sperm DNA Fragmentation Index Techniques, organized by FOG webinar, Advancing Academic Excellence in Fertility, Obstetrics and Gynecology in association with Fertility, Obstetrics, Gynecology Continuing Education Society, Chennai, and also A4 Hospitals and Fertility Center, Chennai. So it's my privilege to introduce the moderator of today's webinar, Dr. Priyanka ma'am. Ma'am is a fertility consultant at A4 Fertility Center, Chennai, and ma'am has more than 10 plus years of experience in the field of fertility, gynecology, and obstetrics. Ma'am has presented many papers at national conferences and commendation received from the Ministry of Railways, Government of India during COVID times. I welcome you, ma'am. And it's my honor to introduce Dr. Jairaman sir. Sir is the senior embryologist, Master of Science in Biotechnology in Annamalai University 2010, and Master of Philosophy in Biotechnology in Madras Veterinary College. And sir has worked as a research project fellow in UGC and published partial 16S RNA of 2 bacterial isolates in NCBI, and sir has more than 13 plus years of experience in clinical embryology. I welcome you, sir, and I hand out the moderators.
Thank you.
Thank you, Tara. Thank you, ma'am. Can I have
Thank you,
madam's CV?
Yes, ma'am.
Hi all, I welcome you to this FOG webinar. It gives me extreme pleasure to introduce Dr. Tara Mahendran. She's currently the Scientific Director in Andrology Center, Coimbatore, India. She's been a postdoctoral research associate in University of Essex, UK. She's been a research technician in National Institute for Medical Research, UK. And she's been a honorary research assistant in University of Leicester, UK. Sorry. Over to you, ma'am.
Thank you very much. I'll just share my screen. Yeah, is it visible to everybody?
Yes, ma'am.
Yeah,
Okay.
ma'am. Audible.
Yes, ma'am.
Okay, yeah. So first I'd want to thank the organizers of for holding this webinar today. And the topic that has been given to me is comparison of sperm DNA fragmentation index techniques. So I'm going to talk a little bit today about what is sperm DNA fragmentation, a little bit about the clinical implications of high SDF, what are the various diagnostic testing methods available out there today, then what do we do in cases of a high sperm DNA fragmentation, and finally I'm going to end with showing you some of the data that we have generated in our lab. So as we all know, infertility is on the rise. Nearly 1 in 6 in the population is facing some form of infertility, and of these the male factor accounts for nearly 40%, and this could be due to varying factors such as where age may play a role, lifestyle, pathophysiology, or even the environment, so exposure from radiation or other things. So coming to sperm DNA fragmentation per se, we often confuse DNA damage and sperm DNA fragmentation. So I just wanted to put it out here that anything which is related to say mismatched bases or dimers, cross-linking, or any sort of base modifications, they all come under the head DNA damage. But when we talk about breaks in the DNA strand, so we could be talking about single strand DNA breaks or double stranded DNA breaks. So this DNA breaks is what we term as DNA fragmentation, and all the tests I'm going to talk to you about today will help us analyze only these DNA fragmentation. We are not looking at DNA damage. So single stranded DNA breaks arise due to oxidative stress or faulty enzyme activity, and they result in a lack of clinical pregnancy or a prolonged time to conception. Whereas when we look at double-stranded DNA breaks, the causes are due to faulty DNA repair mechanisms, which could be brought about due to external radiation or other factors. And what this results in is poor embryo quality, higher risk of miscarriages, and implantation failures. So I wanted to touch upon this. So this one aspect which I'll cover through my talk is something known as chromatin packaging. So in the last part of spermatogenesis, there is something called nucleosomal organization which takes place, where these certain proteins which are called histones, which are associated with the DNA, they are converted to protamines. What these protamines do is they bring stability and they protect the DNA from mechanical and environmental damage. So there could be many different reasons why sperm DNA fragmentation could be caused. So we broadly classify them into testicular and post-testicular mechanisms. So when we look at the testicular mechanisms, we are looking at things like abortive apoptosis, where cells that are due to be have a natural cell death skip that step and then get passed along. We also have things like defective maturation, where the previous slide, this step that I explained is skipped, and we have cells which have immature nuclear proteins. They continue through and are ejaculated. So coming to post-testicular mechanisms, these mechanisms completely are due to oxidative stress. And how does this oxidative stress come about? It could come about due to various factors, from the presence of a varicocele to infections which give rise to reactive oxygen species, to aging, lifestyle factors such as smoking, alcohol, exposure to environmental toxins, and ionizing radiations. So the three main questions that I wanted to address in today's talk, or these are the three main questions that I very often get, which is why I wanted to talk about it today, is when do we test for sperm DNA fragmentation? There's a lot of doubts about whether it's a routine test or only to be done in certain indications. Next, when we have decided to test, then which is the test we choose for sperm DNA fragmentation? And finally, when we get the report, what do we do if the DFI is high or low? How do we explain it to the patient and for our treatment management as well? So coming to when to test. So there are a number of different clinical indications. The most recent 6th WHO manual has included sperm DNA fragmentation in the extended tests. Before, it used to be an advanced where it is not really required, but it has come into extended tests. So it is still not a routine test like a semen analysis. But there are certain cases which I have explained over here below. So it could be in cases of unexplained or idiopathic infertility, previous failed ART treatments, recurrent pregnancy losses, or even abnormal semen parameters along with an associated risk factor. Presence of a clinical varicocele. It can be a palpable varicocele with normal semen parameters, or a grade 1 varicocele with abnormal semen parameters. Advancing male age, if there is certain lifestyle or occupational risk exposures, and also when there is the presence of culture positive sperm. So there is a lot of data out there in the literature which shows that sperm DNA fragmentation testing is indeed clinically relevant. So there's a lot of correlations with poor embryo development, lower implantation rates, higher miscarriage rates, early embryonic development, lower live birth rates after a fertility treatment. This particular review was done in about 2019, where they looked at nearly 1,600 publications, and they were looking at primary and secondary infertility, and the study parameters included changes in semen, various semen parameters, sperm DNA fragmentation values were looked at, also fertilization, pregnancy, birth, and miscarriage rates. So the conclusions from this study showed that in ART conceptions, men who had high DNA fragmentation rates showed embryo development arrest mainly after the second cleavage state. There were decreased success rates and increased miscarriage rates following ART treatments. When it came to varicoceles, they looked at a total of 21 studies, and they saw that there was an average decrease of 8% in the DFI value post about 3 to 12 months after the varicocele repair. Men with a history of recurrent pregnancy loss had significantly higher DNA fragmentation values compared to the control group. In cases of idiopathic or unexplained male infertility, even patients who had normal sperm semen analysis reports showed an average DFI value of 30% or higher. Those are the patients who reported with idiopathic infertility. Then coming to lifestyle exposure, there was lower fertility potential linked to high DFI in cases where there was exposure to radiation, heat, cigarette pollutants, even agriculture exposure to pesticides. Any patient who was serology positive, increased weight, so obesity was another factor contributing to this, increased male age, and also diabetic patients. So coming to the next part, which is which test to choose. So currently there are no sort of guidelines or recommendations by any of the clinical or professional bodies. But these guidelines on which test to use, the guidelines highlight the use and the importance of sperm DNA fragmentation testing, especially in these cases which I have just explained previously. So something that we should consider when we are looking at what DNA fragmentation test to use is looking at the sensitivity and specificity of the test, because determining the accurate DNA fragmentation value will help in the further treatment management. Next, the reproducibility and reliability of the test. So it should be consistent across different laboratories and operators. We need to minimize subjectivity. Also the technical requirements. So is there a need for specialized equipment, technical expertise? Everything should match the available resources. We also need to look at the clinical relevance. So every test has to have its own threshold values. So beyond this, maybe you should go for an IUI. Beyond this value, you should go for an IVF or an ICSI. Finally, what has to be considered is the turnaround time and the cost effectiveness for each clinical practice. So coming to the term DNA fragmentation index, which we commonly call as DFI today, this term was originally coined by the father of sperm DNA fragmentation, Dr. Don Evenson. So he coined this term to define a population of the cells which have moderate to high DNA fragmentation levels. Currently, this term DFI is used by all different testing methods to express sperm with fragmented DNA. However, each different test has a variation in threshold values, and you have to have a standardization of these values for it to be clinically useful to the doctor and the patient for treatment. So I'm going to talk about 4 methods which are very commonly available today to test for DNA fragmentation. So the first method I'm going to talk about today is the tunnel assay. So this assay uses, it can be done on a flow cytometer or a fluorescence microscope, and sperm is mixed with an enzyme called TDT and a reporter molecule called DUTP. So what happens over here is this DUTP is added at the sites of the DNA breaks, and the sperm are then termed as tunnel positive or negative. So this is one of the tests. The second method is the method we do in my lab, which is called the sperm chromatin structure assay or the SCSA test. So in this, a semen sample is denatured by acid. It's mixed with a reporter molecule called acridine orange, and then it is analyzed on a flow cytometer, and the intact double-stranded DNA is labeled green and the fragmented DNA is labeled red. So we look at the difference between green to red ratio of total sperm analyzed. Coming to the next method, which is very popular these days, is something called the SCD or the sperm chromatin dispersion assay. So in this assay, again sperm cells are denatured by acid. There is a staining protocol, and then it is observed on a light microscope. So sperm which don't have any fragmentation and have intact DNA produce a halo, and sperm which have fragmented or broken DNA will not produce a halo. Coming to the last test, which is the Comet assay. In this assay, sperm are decondensed, the semen sample decondensed, mixed with an agarose gel, and then subjected to electrophoresis. So cells which have fragmented DNA, they form a sort of a comet-like structure, which you can see over here. And the high molecular weight, which are intact normal DNA, they do not form this comet, and they look like this. They have an intact, just a halo, round thing. So this is done via electrophoresis. So I just wanted to point out this slide to you. So this is the structure of a toroid associated with protamine and DNA. So this is what the DNA looks like inside the sperm. Now with the tunnel assay, so before that actually, this red part that you can see, that is very tightly compacted sperm DNA and protamine. So it's the protamine toroid, which is very tightly compacted, which you would remember from a couple of my earlier slides. And this nuclear matrix that we see here, it's more loosely coiled DNA, right? So when we are talking about the tunnel assay, like I explained to you, the tunnel assay uses a reporter molecule called DUTP. So this DUTP is a quite large in size. So because of its large size, it is not able to intercalate in this tightly packed toroid region. So any sort of damage that is there in this toroid region gets missed out in the tunnel assay. It is only able to incorporate here these pink things in the loosely coiled DNA part. When we look at the SCSA test, the reporter molecule, like I explained to you, is a dye called acridine orange. It is extremely small in size, and because of its small size, it is able to intercalate into this tightly packed protein toroid region, and it is able to find where there are breaks in the DNA strand. So not only in this nuclear matrix over here, this toroid linker region, which is loosely packed, even in the very tightly coiled protamine toroid region, we are able to find if there are DNA breaks in the particular semen sample. So coming to a comparison between SCSA and the SCD kits. So the SCD kit, over the next few slides, I'm just going to show you a comparison of data that has been done in our lab. So as I explained to you, the SCD kit is a slide-based microscope-based method. Because it is a microscope-based method, it is subject to very high variability, and because of the operator dependency. So actually what we don't realize is there are no clinically valid thresholds specifically for SCD kits. So the threshold values that we see today, like below 30% is normal, more than 30% is abnormal, they have been taken from the SCSA test. So there is no published values for SCD kits as such. The next thing we need to consider is how many cells are we counting. So with the kit, because it's a manual operator dependent, we are counting between 100 and 200 sperm cells. Something that is not looked at is something called HDS, which I will explain in my following slides. It talks about the chromatin integrity within the sperm cell. So this was a study we did in the lab where we got 2-3 different brands. So the market is now flooded with different brands of SCD kits. So as a clinician, which is the one should I choose, which gives me a better, more consistent result? So what we did was we bought 2 different kits into the lab. We had 3 different technicians analyze the same sample, patient sample, same slide that was made. We were quite shocked to see high variability, which is, so each green bar, the green represents one kit, the yellow represents kit number 2, and the red represents the SCSA test. So you can see for patient 1 over here, the SCSA test value was, DFI value was 63%. With kit 1, technician 2 recorded a value of 73%, and it was as low as 35% with kit 2 amongst 2 of the technicians. So such high variability, which test result should you be believing? Because a result of 73% has a very different clinical implication for that patient compared to a result of 35% DFI. So this is something we need to look into while looking at which test we choose. So this was the same study which we published an article on this, and you can see we compared it, these are the values compared between SCSA, Halosperm and the CanFrag SCD kits. With SCSA, a majority of the patients DFI lay in the range where they could go for an IUI procedure. But when we looked at Halosperm, the same patients, the proportion going for an IVF or ICSI increased significantly, and this increased to more than double in the CanFrag kit. So what we are saying is each particular test, how do we know with confidence that we are giving the right treatment to the patient based on the DFI value? So that is something to consider when we are looking at these tests. So coming to the SCSA test, over the next few slides, as requested by the organizers, I'm going to go a little bit in detail about the benefits of the SCSA test and why it is recognized as the gold standard test worldwide. So for one, it is an international test, it has been patented, and there is a standardized report format. So any lab which offers the SCSA accurate genuine test will give you the same report format. And these, you can be assured that the accuracy of this report is extremely high. And like I said, there is something called a DFI and a HDS value. So these are the DFI threshold values as per the SCSA test, and they have been based on hundreds of thousands of samples that we have come up with this threshold value. So as per this studies, we can say with confidence that anything below 15%, we call it excellent to good pregnancy outcomes. So we say that the patients can try for an in vivo or IUI this is without any female factors. That is very important to be noted. If it is a purely male factor and this is the DFI value, this could be the treatment options. As the DFI starts increasing more than 20%, you will see here the graph drops for pregnancy outcomes. So more than about 25 to 30, we recommend moving to an IVF or an ICSI procedure. For values of DFI more than 35%, we see very increased levels of miscarriage and pregnancy loss. So in this SCSA test, this is the first graph that we give in our report, and the x-axis stands for fragmentation, so levels of fragmentation, and the y-axis stands for the green fluorescence. So the x-axis is red fluorescence and the y-axis is green fluorescence. Every single dot that you see over here in this report is a single sperm. So for example, this sperm has high fragmentation, this sperm has even higher fragmentation. So this test measures between 5,000 to 10,000 sperm cells to generate the report. And this DFI is a total of moderate damage and high damage. Why does this affect a clinician? Why should a clinician be bothered about a moderate damaged sperm and a high damaged sperm? So moderate damaged sperm, what happens is they fluoresce yellow on the flow cytometer, and they are capable of fertilizing the egg. So fertilization occurs, but post day 2, when embryo development starts and when you see the embryo arrest happening, that is due to maybe the fertilization by a sperm which has moderate damage. So we see sperm which have moderate damage capable of fertilization, but further embryo development is restricted because of these DNA damage, and the oocyte repair capacity is not enough to repair whatever DNA breaks there are in the embryo. So this is why it's important to separate the moderate damage from the high damage. We don't just give a total DFI value. Again, the sperm to the report is generated based on duplicate measurements. So it's measured twice on the flow cytometer and then the report is generated. So coming to a very important aspect called HDS in this SCSA report, something called high DNA stainability, which none of the other tests give. So why is HDS so important? So what is HDS first of all? So this is a population of cells which have immature nuclear proteins. So in the test, any levels greater than 25% are considered negative for pregnancy outcome. So I'm just put this slide here to remind you once again about how the histones are replaced by protamines. So any sperm which retain these histones, that is what we are measuring in HDS. So sperm which have not converted to DNA plus protamine. Okay. And how is this measured in the SCSA test is, so histone associated DNA, they take up this reporter acridine orange and they stain more than 2.3 times more compared to protamine stained DNA. So that is how we see a different population on the in our analysis. And HDS is negatively correlated with all semen parameters except vitality, with the strongest correlation being with morphology. So morphologically, HDS cells look abnormal. They have larger heads, long elongated heads, things like that. So what does HDS translate to clinically? So high HDS values give you low fertilization rates, there's impaired embryo quality, arrested embryo development, which results in decreased pregnancy rates. And of this, what is significant is the block at the 2PN stage. And data have shown that sperm with higher HDS are more common in recurrent pregnancy loss compared to the controls. So this is something I just wanted to show you, which we have observed in the lab. So this is a profile or a graph of our data that we generate. So like I told you, we call this a cigar shape, this particular profile. So you can see it is slightly broad at the base and it tapers and becomes narrow as it goes up vertically, right? So this is a normal sperm population, which we would see in 99% of the male samples that we analyze in the lab. Now coming to some patients, we very rarely see some patient samples which look like this. I'm sure you will all agree with me that there is a difference in this cigar over here. So compare it to this where we have a broad base and tapering at the top, and compare it to this abnormal sample which is narrow at the base and it gets broad at the top. So what this means is, this is indicative, or it is a sort of a marker for diploid sperm. So if you look at it, like I explained to you here, this histone DNA, associated DNA, takes up 2.3 times more of acridine orange, which is why this population here is the high HDS population. So these particular patients, they will have normal DFI values. In majority of the cases, they will have normal. So you can do a semen analysis, you'll do a DFI test, they'll have normal DFI values. You'll take them for a ART treatment. It will end up in probably, it won't be successful. Reason being, because of this high HDS value. You can see in this particular patient, DFI is 28, borderline abnormal, but the HDS is 67. So the cutoff for HDS is 25, but this patient it's 67, and this patient DFI of 17, which is considered normal, but HDS of 70. So with the SCSA test, you can do an initial screen of like whether the patient has diploid sperm. To confirm it, obviously, we will need to do a sperm aneuploidy test to check whether really are there diploid sperm in that patient's sample, which will help in further treatment management. Again, these are just more examples of diploid sperm that we have come across in the lab. It's a very rare occurrence, but it is something that happens. And clinically, when we get patients where, DFI is normal, analysis is normal, what could be going wrong? Why are they not having positive cycles? This could be something that should come to mind. So finally, coming to the last part of the talk, which is what do you do if you have a report of a patient which has come back with a high DFI? So there are a lot of management strategies out there, ranging from lifestyle modifications to weight loss. Frequent ejaculation, so reduced abstinence, is something that's becoming very popular these days because it reduces the DNA fragmentation levels. If there is any sperm positive, like it's positive for culture, we treat them with antibiotics. Antioxidant supplementation, very, very important. If there is a presence of a clinical varicocele, to repair the varicocele, come back after a few months and then go for the cycle. Sperm selection techniques and use of testicular sperm. So this is a publication which has shown that across the different intervention strategies, what is the relative reduction in DFI values? So something like a short abstinence could result in an average of 25 to 22% reduction in DFI. Something like a PIXI can result in maybe nearly 60%, which is extremely high reduction in DFI values. and testicular sperm, use of testicular sperm over ejaculate sperm, has very high reduction in relative DNA fragmentation values. Something I'm going to touch upon today is the sperm selection methods, because I feel that is becoming more relevant as we are progressing. What do we do to offer better sperm to the embryologist for a better successful outcome cycles? So these sperm selection methods range good old density gradient centrifuge, which has been used for many years. But coming into the market slowly is Max. It's been around for many years. Microfluidics, the pixie dish, and something called this Felix or electrophoretic separation of sperm. So microfluidics, I'm not going to go into explaining each of the methods. I'm just going to show you some data that we have generated in the lab. So this is data that we have shown where we looked at the semen samples before and after processing with microfluidics. We looked at about 85 samples and we saw an average reduction of nearly 22% across all these 85 samples in the DFI value. And in HDS, there was an average reduction of about 7%. There were only 5 cases where DFI did not increase, but it increased after the use of microfluidics. So coming to this device which I showed you over here, it's called the Felix device. It is an electrophoretic separation of sperm. So it's a device that has been invented by Dr. John Aitken in Australia, and we were very fortunate to be part of this worldwide study to analyze samples in India. So in India, as part of the study, we tested 38 samples and we looked at the raw sample, we looked at density gradient, and we compared it to this Felix instrument. So the Felix, as you can see over here, Felix instrument produced sperm with extremely low DNA fragmentation compared to density gradient centrifugation. The volume that we recovered was extremely low. So that is something they are working on right now because it can be used only for an ICSI and not for an IUI at the moment. But comparing all other semen parameters, Felix was better or comparable to DGC, as I said, except for the sperm concentration. This is another study that we did in our lab specific to the Indian population. So a lot of the time we see studies with DNA fragmentation that have been done in Western populations. I feel since we have such a huge patient pool in India, we should be looking at data from our own country where we are able to show what works and what doesn't work in our population. So this was something that was very special to us, where we looked at nearly 7,000 patient samples and we analyzed age and compared it to DNA fragmentation. So I think it's becoming very common knowledge now that advancing male age plays a very important role in sperm DNA fragmentation. So over here, what we were able to identify is that in Indian males, 36 years of age was a sort of a threshold value. Beyond 36 years of age, significantly the DFI started increasing. So when we get patients in the lab, we say, yeah, okay, more than 40, yeah, we'll look into a DFI. Maybe 30s, not necessary. But this is something that's really important, which I wanted to point out to all the clinicians. So this again is some other data from the lab. This is looking at about 36,000 plus samples. So in the SCSA test, anything more than 25% DFI is considered abnormal. So nearly 4 out of every 10 samples that we are getting in our lab have high DFI and are considered abnormal for sperm DNA integrity. So coming to the relationship between count and sperm DNA fragmentation. So this data is based on about 33,000 patient samples that we have tested in the lab. And something that is normal and would be expected is oligozoospermic samples are very high in sperm DNA fragmentation. So that's not surprising for us when we see that in the left panel, 54% of oligozoospermic patients had high DFI. But what was really surprising to us was 35% of normozoospermic. So a lot of the times we do a semen analysis and we stop at that. But 35% of those patients who report with a normal semen analysis actually have abnormal DNA fragmentation. So these are the patients who are being missed, and then further treatment options are not, then we are not considering the right treatment options because we are overlooking this DNA factor in the sperm. So finally, to conclude today's talk, I hope I've given you enough data to show that clinical utility of sperm DNA fragmentation is proven in evaluation of the male, and it's also, this DNA integrity is very crucial for fertilization and health of the offspring. So high DNA fragmentation values of impacts embryo development, implantation, and natural pregnancy rates, and pregnancy rates in natural and ART conceptions. The choice of method is extremely important, and the clinical indications need to be considered. For clinical use, using the right diagnostic threshold values according to the testing method has to be considered. And finally, coming to treatment options, which include antioxidant intake, shorter abstinence periods, varicocelectomy, lifestyle modifications, use of testicular sperm, and advanced sperm selection methods. So thank you for your time today, to this patient listening for the talk.
Thank you, ma'am. Thank you for excellent, elaborate presentation for this session, ma'am. All the participants, you can put your questions over here, so we can ask the question to the center.
We've got a
Ma'am,
question, ma'am. So, is there a cutoff of high DFA above which we should absolutely go for TISA? Like for a, so that's the first question.
Yeah. So, see, nothing is absolute, right? We know in this infertility field there is always a case of, yeah, there are higher probabilities of success. But what we advise our clients is beyond 45 to 50% of DFI, a TISA sample would be probably, it would have better outcomes than an ejaculate sample. That is as long as the HTS value is normal. So if the HTS value is normal, above 45%, we recommend you go for a TISA, but not absolutely. It depends on all the factors considered.
For a previous ICSI failure with cleavage arrest, high DFA with microfluidics, how to proceed with next cycle, ma'am?
So it's already been, microfluidics has already been
In
used.
trying, yeah.
Yeah. Okay. For that, once again, then I would look into what is the cause of the DFI. So if there is something, so this microfluidics and whatever we are talking about, sperm selection, they're just immediate, it's a immediate thing where we are just reducing the DFI and then using it. But if there is some particular problem in that, which we can probably cure with a slightly longer course of treatment. So is there some sort of exposure to the patient? Is there a varicocele? Um, is there some culture positive sperm? So if we look into all that, maybe those can be addressed and then come back to an ART cycle once again, maybe with the use of testicular sample once again.
I mean, certain patients, the DFA still remains high and we proceed for ICSI normal. So, what is the procedure you prefer, ma'am, in such cases of high DFI? Like MAX or ICSI or microfluidics, like whichever?
So we don't. So actually, it's mainly the clinicians' call on that, doctor, because we just deal with this side of it. What I'm trying to establish actually with our clients is, to have a crosstalk where what works for them, what doesn't. So if there is a high DFI, how many cases of that have been successful with which particular type of intervention. So something like that would be helpful. Right now, we test the sample, we give the reports to the clinicians, then we don't hear back whether what happens, whether the cycle outcomes have been positive, negative, live births. So that is something we are working towards. But just from the literature, then yeah, we look at the cause of the fragmentation, if it's something that can be addressed, and then in those cases when even sperm selection methods have not worked, maybe look at use of a testicular sample.
The next question is, please explain microfluidics in short.
We don't use that in my lab, and I am not probably the best person to explain it. I'll just it works mostly on the principle of motility and chemotaxis. So it is the ability of the sperm to move through very micro channels which mimic uh, the the in vivo conditions. So based on that, sperm which are able to swim faster and go in through these channels and come out in the recovery well, that that in short is how these sperm are, the sperm. And the reason why those sperm, they say the sperm have intact DNA, which is why it's able to move like this and come into the recovery well.
The next question is, how long treatment is needed to reduce DFI in your experience? Have you seen?
A minimum of 3 to 6 months, sir. So we don't recommend anything shorter than that. Definitely 3 to 6 months. We've seen some cases where there is a definite. Um, I'm just bringing up. I have a slide where we have some. I can just share. So this was a case where we saw. So male was 40 years. The 3 years primary infertility. They had a history of failed IUIs and IVF. Surprisingly, his semen analysis was normal, but he was a heavy smoker, regular alcohol consumption. So prior to when we took his DFI first, it was 45%. He stopped smoking and he quit alcohol. So 84 days and 4 months. And post that, he came back to retest, and we saw a decrease of nearly half. So decreased to 24%, after which they went for the cycle. So yeah, this was antioxidant intake. So once again, antioxidants for a 3-month period this was. So again, the 4 years primary infertility, 4 failed IUIs, no abnormal lifestyle factors, so non-smoker, no surgical or significant illness. So he took antioxidants for a 3-month period, and the DFI dropped from 52 to 24%. So these are cases where, this is just one case that I've put, but in general we see cases with where 3 to 6 months is the time that we before it thing.
So, which is the most common sperm selection technique in India?
Currently, I think density gradient centrifugation, given the cost and things involved, mostly in all centers it's DGC being used. I think that is slowly moving to microfluidics because uh, again, once again, the utility, it's very easy to use microfluidics. It's a small slide, it's not invasive or anything. Cost is also, for the patient, the end cost is not very significantly higher, and the time taken, when you compare it to Pixie and Max, the cost is extremely high, which is why probably I would say density gradient and microfluidics are very commonly used currently.
What about Pixie, man?
Pixie, sir, I feel cost is a sort of a deterrent, but I'm not sure right now whether the cost is coming down, which is making it more available for clinics. So this is all I should get data from your side, being in the clinical side and the lab side. So do you feel amongst your colleagues that Pixie is being used very highly now, sir?
Yeah, yeah, ma'am. We are using what A4. We are
Okay,
using, yeah,
okay,
so highly
okay.
up to 30,
High.
40% patients. We are using Pixie.
Okay,
That's
okay.
why we
So
use, because you showed that.
cost is not a factor.
No, ma'am, it's
Okay,
hardly 10 to 15,000 rupees, because
okay,
the patients are
okay.
spending too much, though. So we are
Yeah.
counsel the patients regarding that. That's why
Okay,
I just
okay.
asked you. You showed that on slide,
Yes,
Pixie TFI is less like that. You showed, that's why I asked.
yes. Yeah, 60% less according to that study. Yeah, Pixie has shown.
Okay. Ma'am, do you have any significance of single strand break and double strand break on DNA fragmentations? Any clinical significance is there for a single strand break and a double strand break, ma'am?
Um, I explained in the initial slides about what the um, what it results in, but there is no sort of study which is showing that a single stranded break could result in a particular. So the first slide where I showed single stranded breaks result in a lack of clinical pregnancy itself or a prolonged time to conception, whereas double stranded breaks you will get fertilization, but then embryo quality is very poor, so you have a higher risk of miscarriage and implantation. So in a lot of cases, if we are seeing fertilization happening, but then implantation and miscarriages, implantation failure and miscarriages, that could be due to the double stranded breaks. But in terms of a particular test to distinguish between single and double stranded, only the comet assay will be able to do that, and that too alkaline comet and a neutral comet. So one comet will be able to distinguish single stranded DNA breaks and
Okay.
one will be able to double stranded. But clinically, the only data we have is lack of clinical pregnancy and then embryo quality implantation failures with double stranded breaks.
Okay. So, what are the equipments needed for SCSA techniques, ma'am? Can you elaborate on that?
Yeah, so we require a flow cytometer for to do the SCSA test. It is a flow cytometer, and the actual SCSA test, it's a licensed test, so it is given only to certain authorized labs. So you require a flow cytometer and the software. So the software is what is done on a franchisee basis. So for India, we are the only lab who has the franchise for SCSA in India. So this software is required. So we upload the data onto that, the raw data that's processed on the flow cytometer, and then you get the standardized report format. So anywhere in the world, if you go, you get that same SCSA report format. So the machine is sort of modified. So there are certain modifications that we do to the flow cytometer before using it for the SCSA test.
Then I can understand, ma'am. Okay. So, if practically we are doing is TA method right now. So, do you have any suggest us if there are any cutoff, then we can send the samples to
So
you.
which method, sir, are you doing? Sorry, I didn't hear.
Ma'am, SD means sperm. Sorry, STD.
SCD,
Yeah,
the
sorry,
kit.
STD.
Yeah, the kit. Okay, yes, sir.
Kit
Okay,
kit
okay.
method, ma'am.
Yes.
So we are
Okay.
using that only. So
Okay.
if is there any problem or errors if you are finding, so we want to check once.
Yeah.
So
Okay.
we want to send that sample to you, right? Do you suggest us or recommend us any cutoff values if
So
there any
more than the
is there?
cutoff value, sir, I would recommend on the patient history itself. If with the SCD method, you're getting a normal value, but the patient is coming with a history of failures, so either miscarriages or previous ART failures, those samples could be sent to us where you can check whether HDS is abnormal for the presence of diploid sperm, or you can check the ratio of moderate to high damage. So even cases where DFI is looking normal or high in the kit method, but you're getting fertilization, but after that from day 2 the embryo start, there is arrested development. So day 2 onwards, arrested embryo development, those could be cases to send the semen sample to us. If you see a higher range of moderate damaged cells, those cells could be getting fertilized, but then post day 2 embryo development is stopped. So those are the cases. More than a cutoff DFI value, I would think it is more on the type of case that we see.
So history of the
Then send.
patient history.
Yes, sir, correct. More than the cutoff value.
Okay, ma'am, I understood. Then you showed that even 35 percentage of normal normospermia patients have high DFI.
Yes.
So from that point, I get a question that whether you are recommending DFI for all the patients, is it needed, ma'am?
So, like I said, the guidelines and none of the professional bodies are advocating for routine testing. So, it is dependent on the clinician and the center and what SOPs they have come up with. So, a lot of our clients do routine testing. Every patient who comes, they test the sample for DFI. But there are a lot of clients of ours who only indicated cases. They will only send some cases to us. You run the risk of, like I said, that 35% you're losing out on knowing. So actually, at the end of the day, it's the clinician's call whether it's worth the amount to the patient to do the DFI testing to get a full investigation before proceeding with the treatment, or whether if it is an economic burden for the patient, then maybe you go for one cycle and then test the DFI. So I think that has to be weighed up in each case.
Yes, ma'am. Yes, ma'am. Usually, the indicative patients only they are doing for DFI.
Okay.
That's why you
Okay.
showed
Even
the
you are
data.
only
That's why
doing, yeah.
we are also the same only. But we are looking forward whether we have to incorporate our protocol to do or DFI for all patients.
All
We
okay.
are under sort of confusions right now. That's why I just asked the question.
Yeah,
I think you are
it
the right
varies.
person.
It varies. So, lot of our clients, like I said, some centers, which send every sample they send to us, but some centers they pick and
Okay, okay.
choose only based on the indication. So, I don't know what the reasoning is for that. If it's a financial reasoning or whether they don't want to just burden the patient with unnecessary test or yeah,
Usually the financial
yeah.
we take and apply.
Any
Any
questions
more
from the
question,
audience?
Priyanka ma'am?
Was there any questions from the audience? No, sir. Thank you, sir.
Thank you, ma'am.
Thank you, ma'am.
Ma'am,
Okay.
Thank you,
it was an
Tara. Thank
excellent
you for the wonderful
presentation,
session.
ma'am.
Thank you.
Thank you very much for giving us this chance. Thank you so much.
Thank you. Thank you, sir.
Thank you,